my name is Melanie, and I'm part of the Go-Getters. Today we'll be talking about a method called CryoEM and how it's used to determine 3D protein structure. So first we'll talk about the history of 3D modeling. So transmission electron microscopy, or TEMs, utilize an electromagnetic lens, which creates an electromagnetic field to focus electrons into a fine beam. It doesn't use a glass lens like normal microscopes, and the beam passes through a thin sample, causing electrons to either scatter or hit a fluorescent screen at the bottom of the scope. After the electrons hit this screen, it projects an image of the sample onto a detector, which is shown in different shades due to different electron densities. Therefore, TEMs require a very thin sample that has to be less than 100 nanometers in thickness. These images are shown in different shades due to different electron densities. For example, a sample with less density would cause more electrons to get through, causing a brighter image, and vice versa. So because electrons have a shorter wavelength than light in normal microscopes, it results in a higher quality/resolution of images than previous methods. TEMs are used in scanning, diffraction, and spectroscopy. However, there are some disadvantages to this method, as some samples like biomolecules are too fragile for powerful methods like TEMs, which utilize intense electron beams. The water surrounding these molecules evaporates, burning and destroying the sample. It's also incapable of analyzing certain samples because of the electron irradiation, which damages samples even further. After TEM proved to have some disadvantages, Xray crystallography was developed. Xray crystallography analyzes the diffraction from a beam of X-rays. These rays are redirected with a crystalline atom, and by measuring the angles and intensity of these beams, scientists are able to produce a 3D picture of the density of electrons within an atom. And using these values and this data, they're able to determine positions of atoms in a crystal, chemical bonds, disorder, and other properties. The only thing with this type of method is that it requires a sample to be crystalline first, which isn't always possible and can be very difficult to acquire. This type of crystallography enabled big advancements in biology, such as when Dorothy Hodgkin won a Nobel Prize for solving structures like penicillin, and the structure of DNA was also revealed using this method. So this method is not always possible due to the fact that we can't always get a crystalline sample, and it also takes extremely long. The problem. Xray crystallography contains a few problems. First of all, the picture becomes much fuzzier and lower quality as the item becomes more complex. It's extremely hard to get proteins to crystallize into a regular structure, unlike their natural complicated arrangement. And by placing these samples in non-physiological environments, it can denature the sample or just change its function. Another issue with Xray crystallography is that there's no way to generate 3D models, and we're only getting part of the picture. It also fails on proteins that move or are fluid. If proteins are moved during the imaging process, the image would blur, and Xray crystallography would not be able to take an accurate picture. Later, another method called CryoEM, which we'll be talking about later, solved this by first freezing the proteins to prevent movement, and then using an electron microscope to get an image at a higher resolution and a smaller scale.
So one of the big advantages of CryoEM, which we've probably already talked about, is the ability to image non-static proteins, or proteins that tend to move around a lot. We do this by freezing the proteins in liquid ethane, then taking that frozen grid and storing it in liquid nitrogen, which is at about negative 210 degrees Celsius. Once the particles are frozen in place, we use TEM, or transmission electron microscopy, to capture shadows of the particles, and then take those 2D shadows and reassemble them into a 3D picture using a computational algorithm. So in a bit more detail, the way an electron microscope works is it shoots a beam of electrons at the proteins, and the organic molecules in the proteins block those electrons, so the sensor at the bottom of the microscope only captures electrons that weren't blocked by the protein, essentially getting the shadows. One big problem with using an electron microscope is that the radiation could damage the proteins. So we use something called single particle electron microscopy, which minimizes the amount of radiation that the proteins are exposed to, keeping them safer. We reassemble these 2D images using subtomogram averaging. A tomogram is just one of these 2D sets of images that we get from microscopy. And once we analyze and align them correctly, we can reconstruct them into a 3D model.
In order to solve the problems posed by Xray crystallography, 3 scientists rose to the challenge of finding new ways to innovate the field. The first of which was Richard Henderson, a Scottish biologist. He worked with transmission electron microscopes at first, but was interested in developing 3D shapes. He did so by taking 2D images from multiple angles, and then he combined them into making one 3D shape. This was very effective. However, the transmission electron microscopes were not precise enough to make great 3D structures. He then proceeded to refine the transmission electron microscopes and make more robust 3D images, which allowed him to reveal that most biological molecules and proteins had their own robust structures, such as alpha helixes and deep-plated sheets. The next person to help in the process of finding a new way to model 3D proteins and biological molecules was Joachim Frank, an American biophysicist who was born in Germany. He was working at Columbia University and took Henderson's method and made it computational. He did so by using a computer to analyze the 2D images instead of by hands, and this allowed him to make sharper 3D images and do so faster and more effectively. Finally, Jacques James Dubashe put the cryo in CryoEM when he developed the method for freezing ethane. This method was so important because it vitrified the liquid, which means preventing crystallization and making it one solid homogeneous glass surface. By doing so, it allowed proteins that couldn't be crystallized when retaining their natural shapes to retain their natural shapes and be able to be photographed from multiple angles. This allowed many more different kinds of proteins to be analyzed and made the cryo-EM method so much more lucrative.
Henderson, Frank, and Dubashe, who were mentioned previously in this presentation, were awarded the 2017 Nobel Prize in Chemistry for their role in the development of cryo-EM, which really shows how important this technology is. This was awarded 27 years after Henderson's initial publication of a protein using electron microscopy. Applications of cryo-EM and the 3D images they produce include finding the function of a protein because the shape of a protein is important to what it does. Discover the shapes of molecules not able to crystallize as you try to crystallize molecules before you image or picture them, and study protein activity, which can help with drug design because some proteins are used to deactivate or activate other proteins, and knowing the shape of those proteins can help with this process. Some pros of cryo-EM are smaller samples are needed than previous imaging techniques. It's fast for simple structures. The crystallization process preserves the hydrated state of the sample. It has more versatility and greater distinguishment or higher resolution images. The cons are low contrast. The more complex a protein or shape gets, the longer you have to wait, and sometimes it can take hours or days for the data to process and the 2D images to be mashed into 3D images. It's costly, but there's hope that the price will go down, and you can't photograph certain angles.
In order to get a 3D image from a set of 2D images, the first step is to perform a Fourier Transform on each of the 2D images. A Fourier Transform converts a function into the sum of its sine and cosine components. Then the Fourier Transform of each of the 2D images is combined, and this helps form a 3D image. Cryo-EM machines can be very expensive as they can cost up to $17 million US. However, the average cryo-EM machine costs only around $5 to $7 million. In addition, they cost a lot of money every day to run, and they require specialized labs that minimize vibrations. This is because vibrations in the lab can cause blurring of the image and decrease the resolution. Thus, many major research areas can afford cryo machines. However, many other smaller research facilities without the necessary funding for a cryo-EM machine cannot afford to buy them. Thus, they can instead get a cheaper alternative, such as a 100 to 200 or 200 kilo electron volt microscope, which costs $1 to $2 million, which is much less than the normal cryo-EM machine. Due to these high costs of CryoEM machines, people are researching different ways to replace their function by a less expensive machine. For example, in March 2020, researchers at the Okinawa Institute of Science and Technology made an imaging scanning electron microscope, and this is simpler to use and is much cheaper than the CryoEM machine, and it also uses lower energy electrons, which can help improve the resolution. And in the future, we have a lot of room to improve upon our cryo-EM and other techniques for imaging of cells and other biological specimens, and this can help decrease the cost and increase the quality of our results.